Well, good morning, welcome, and thank you for attending our webinar. So I will discuss the various tools in our Admet Design Suite and show examples of how they were applied to an in-house project to discover antimalarial compounds. The next slide is simply an outline of the presentation. So first I'll discuss our strategy for designing potential antimalarial compounds, and this will be an overview of the design process. I'll be coming back to this slide over and over again to when I come to key points within the talk. Of course, we build a large number of models, and so we created a model building process or flow chart, which I'll illustrate in the next section. We have more than 140 predictive models in Admet Predictor, and they were basically built using this flow chart. Next, I'll discuss Admet Risk, which is an Admet liability score. We have a large number of models, and the Admet Risk allows us to kind of summarize all these models into a simple score. Then I'll discuss Medchem Studio and the class generation method that was used to during the design of the antimalarial compounds, and I'll also discuss R group explosion, which allows us to generate novel analogs by recombining substituents. Then in the final section, I'll talk about the results of the compounds we synthesized, their biological activity in the antimalarial assay, and also the Admet type properties of these compounds, which we also had tested so that we could compare the predictions to the actual experimental results. Okay, so GlaxoSmithKline screened about 2 million compounds in a single assay, a single concentration of 2 micromolar, and a parasitic assay that contained red blood cells. So they were looking at growth inhibition of the cells. They made available 13,456 active compounds where the percent inhibition was greater than 80% at 80 micromolar. Since it was a cell-based assay, the mechanism of the compounds is unknown. So what we wanted to do was take literature antimalarial enzyme targets, create QSAR models of those compounds using our Admet modeler software, and then predict the activity of these 13,500 compounds with our QSAR model. We also computed Admet properties of all the compounds using Admet Predictor. The next step was to classify the molecules into various chemotypes, in other words, group the molecules together using Medchem Studio. Then we looked through the various classes and identified attractive structural classes. So these would be classes that were potent, had low Admet risk, and attractive synthetically. We then used the R group explosion to generate novel compounds, then reapplied our QSAR model to the new compounds, and also applied Admet properties, and then filtered based on Admet risk. And when we got down to a manageable number, we simulated the dose necessary to achieve the therapeutic index using GastroPlus. So this ruled out a few of the candidate compounds. And then finally, we had a small number of compounds which we had a contract research organization synthesize, and then we had tested in a biological assay at the University of California, Riverside. Okay, so this slide illustrates our overall model building process. So this is the process we would use to build the 140 predictive models in Admet Predictor. So we start with with structures and experimental data, and we spend a time curating the data. If you don't have good data going into the model, you're not going to be able to build a very robust predictive model. The next step is to compute molecular and atomic descriptors, and we have over 330 descriptors, and they're broken up into these 6 classes. So now you can think of a spreadsheet where the rows are the molecules and then the columns are these various descriptors of the molecules. And what we're going to try to do is build a mathematical equation that relates the activity or the physical chemical property of these molecules to the descriptors. The next step is to remove low variant and correlated descriptors. So if we have a particular descriptor that's not varying from row to row, it's not going to be helpful in differentiating the biological activities of the compounds. Similarly, if we have 2 columns that are highly correlated with each other, we'll drop one of the columns so that we don't introduce redundancy into the descriptor set. The next step is to create a test set. And so we typically take 10 to 20% of the compounds in the data set and sequester them in a test set. And then when the model is produced, we apply the model to the test set. And there are various ways listed here to create the test set. So the 80% or so compounds that are not in the test set are used as a training set and introduced into an artificial neural network training to create various models to predict the endpoint. A priori, we don't know what the number of descriptors or neurons will produce the best model. So we create a grid of artificial neural network ensembles by varying the number of descriptors and the number of neurons. So each one of these cells here represents a particular architecture with a particular number of descriptors and neurons. Then once the models are built, we apply them to the test set. And this grid allows us to compare the predictions on the training set to the predictions on the test set and those statistics. If you're getting very good predictions of your training set, but poor predictions with your test set, that may mean that the model has been overtrained. And we go to great lengths to not overtrain the model, because if you overtrain the model, you're going to get good results in a training set, but not be able to predict outside molecules. Then finally, we select one of the architectures, and that becomes our artificial neural network ensemble model for the predictions. Okay. So there's 2 types of models we create. One is a classification model, and the other type is regression model. So on the left is a classification model. Here you're just trying to classify the molecules either as non-toxic or toxic, or maybe a substrate for a particular step or a non-substrate for a particular step. So what you get out is predictions of the compounds, and down in this lower left-hand corner, these are compounds that experimentally are non-toxic and that we predicted to be non-toxic. So we have these correct, and then similarly in the upper right-hand corner, these are compounds that we predict to be toxic, and in fact they are toxic. And then in these 2 corners, these are compounds that we incorrectly predict. So here we predicted these compounds to be toxic, whereas in reality they were non-toxic. So a number of statistics we get out of the model that we analyze. There's things like specificity, sensitivity, and false rate. Then over on the right-hand side is an illustration of a regression model. So this is probably more common to most people, where you have experimental values and you're simply trying to create a model to predict their activities. In this case, a log P, so it's a physicochemical type property. And as you can see, our log P model correlates very well with the observed experimental values. And one of the reasons is we have a very large test set of these compounds. So we have, a data set. So we have 1,258 in the test set and over 11,000 molecules in the training set. And as you can see, we have a very good R-squared correlation between the experimental and the predicted values. Okay, so going back to our design process, our overall strategy, the first step is to create QSAR models of potential anti-malarial targets. So there were 2 literature targets we looked at. One was cyclin-dependent kinase for the parasite, and the other was dehydroorate dehydrogenase, or DHODH. Okay, and so I'd like to illustrate how Admet Predictor can quickly create a model of DHODH. So this is our Admet Predictor program, and I've opened up a SMILES file of literature DHODH inhibitors. And I have a column of the inhibition constant, the Ki value, in micromolar quantities, and then I've converted that to minus log of the Ki value. So for if you have a pKi of about 8, that would indicate a molecule with a potency of 10 nanomolar. Okay, we can sort the compounds based on the pKi. So this is the least potent compound in the data set, and then this is the most potent compound in the data set. Okay, we can also quickly look at a distribution of the activities. So they go from a pKi of 4 to about a pKi of 8. So the first step in the process is to calculate the descriptors. So I simply go to the Calculate pull-down menu and select Calculate Admap Properties. So this very quickly calculates the 330 descriptors for these 89 molecules, and also calculates various physical, chemical, biopharmaceutical, metabolism, and toxicity. So this has calculated all the predictive models we have. And these tabs down here let us see various properties. So here's our physical, chemical, and biopharmaceutical models. Here's our metabolism models and our toxicity models. And I'll talk more about those later on in the presentation. Okay, so the next step is to define our dependent variables. So this is what we're trying to predict. So in this case, it's the pKi of the Plasmodium falciparum DHODH. Here we have over on our right is the descriptors will allow the model to train on. If we wanted to add some of these descriptors on the left, or if you had your own proprietary descriptors, you could simply select these and click this button, which would transfer them and allow them to be available for training. Next thing I need to do is select the definition of the test set. So I'm going to use a minimum test set size of 20%. So I'm going to have 20% of the compounds going into the test set. And I'm going to define these as every nth compound sorted by the pKi value. So this allows us to have an even distribution of pKi values in the test set. Next thing I need to do that I haven't talked about previously is select how we're going to narrow this large number of descriptors down to a particular number for an architecture. And there's a number of different methods: input gradient, truncated linear analysis. I'm going to use a method called genetic algorithm, and it's going to apply this genetic algorithm to each one of the architectures in order to try and find the best descriptors for that architecture. Then finally, I click on the AN and E tab, and here's where I'm going to specify the range of neurons and descriptors to which to build models. So I'm going to say go from 1 to 5 neurons and a step of 2. So it'll build 1 neuron models, 3 neuron models, and 5 neuron models. Then as far as the number of descriptors, I'm going to start with a minimum of 5 descriptors, go up to 15, and use this step of 1. Okay, so now I'm ready to start building models. I simply go back into the Basic Modeler tab, click on the Artificial Neural Network Ensemble button, and we're starting to build various models. So while the models are building, let's briefly look at the log file to show what's going on. So in this first section, we're eliminating descriptors that have very low variance. So these are descriptors that won't be able to differentiate the activities of the molecule. So these all, essentially, there's no compounds with phosphorus, so all the rows for that particular column are 0. Then if we look further on down, here's where we're looking at pairwise correlation between the descriptors. So for example, molecular weight is highly correlated with number of electrons, so we would drop one of these 2 descriptors. So now let's go back and look at our model building process. So now we're at about 10 inputs, and it's going very quickly through these various models, as you can see in the GA steps down here. And we should be finished in a matter of seconds. And as you see, the training is complete. So now we can simply click on this button, find the best ensemble, and it selected a 3-neuron, 7-input model as the best model. And if you look at the statistics, we're looking at the root mean square error. For the test case is 0.53, and for the training set is 0.54. So those are similar, so we're not really over-predicting. If we click on a cell, we get the observed versus predicted plot. We can look specifically at the training set molecules, and they don't differentiate too far from the best fit line. And similarly, we can look at the test set, and we can click on a particular point and see how well we did on the prediction. Here we predicted the activity would be a 7, and it was actually 6.4, so not too bad. So if we had decided that this was our best model, to export it, we simply hold down the control button and click on the cell. That exports it to our Model Export tab. Then we can simply click on Export the Current Model, give it a name. I'll just call it Mike PFDHODH. Click Append as the last model, and then it would show up down at the bottom here, and then click OK. Okay, so now this model would be available for any future runs in Admet Predictor, and we would be able to use it to predict PFDHODH activity in Medchem Studio also. Okay, so now going back to the slides, the next step would be to predict the target activity and the Admet properties for these compounds. So we developed the QSAR model here that's predicting our target activity. The next step is to predict the Admet properties. So what kind of models are in Admet Predictor? So we separate these by physicochemical, metabolism, and toxicity modules. This shows some of the models in our physicochemical and biopharmaceutical module. So first we have our Pka Model, which is a multi-product model including all microspecies. Solubility model. First, the initial thing is to predict the solubility in pure water at native pH and a temperature of 25 degrees C. If the compound contains ionizable groups, then the pH will be something different from 7, so we also predict that. Intrinsic solubility, salt solubility factor, and then various solubilities in simulated gastric fluids. We also have a very good log P and log D model, various permeability models such as human effective permeability in the jejunum, MDCK, blood-brain barrier and corneal permeability, diffusion coefficient in water and molar volume, and then various pharmacokinetic models such as human plasma protein binding, and then finally an OATP1B1 inhibition model, which is a classification model. For our metabolism models, these first two relate to is the molecule able to inhibit, or is it inhibiting one of the cytochrome P450s, and these are the major, 5 major cytochrome P450s. Next we have a substrate model. So we classify the molecule as a substrate, yes or no, for 9 different isoforms. If it is a substrate of these, then we can predict which sites are likely attacked by the various cytochrome P450s. We also calculate or predict Kn and Vmax values for 5 cytochromes and also clearances. And then finally, glucuronidation sites or classification model, yes or no. Is this compound a substrate, for example, for UGT1A and these other UGT enzymes? Our toxicity module contains predictive models for estrogen receptor and androgen receptor toxicity, carcinogenicity in rats and mice, Ames mutagenicity models, hERG model, phospholipidosis, reproductive toxicity, and etc. Okay. We've had a number of independent researchers compare various models put out by different software, and that's what we'll discuss in this section. So the first independent comparison was of log P predictors. So it was a paper by Dearden, and he compared various log P predictions to experimental results. And as you can see, we came out on top. So QMPR plus was the old name for Admet predictor. The second independent comparison was performed by Tetko et al. And again, you can see that our model for log P came out ranked 1. So whenever you see an S plus in front of a model, that stands for simulations plus. As you can see also, the A log P model was ranked equally with ours, and this is the model built by Tetko. And then the third independent comparison of log P predictions was performed by Manhold and Tetko. And again, you see that we ranked number 1. Here, this was on a very large data set of about 96,000 compounds from Pfizer. And then finally, comparison of solubility predictors. So solubility is a very important property, but it's somewhat difficult to predict. And as you can see from this table, for Dearden, we came out again on top as far as solubility predictions. So let me just pop into Medchem Studio and just show a few of these properties. So this is a subset of 1,000 compounds out of the 13,000 compounds that GSK donated. And so I showed before how these are in various tabs in Admet Predictor. With our Medchem Studio software, we can simply read in the compounds and click on the Admet, on the AP button, and this will go out to Admet Predictor and calculate the various properties. And also under the customized properties here, it will also calculate and display any user-defined models. So this would have been an example of the PFDH model that I entered into Admet Predictor before. Okay, so here I just simply show the water solubility for these molecules. If you hover over a particular column, it will give you some information about what that model is. So this is water solubility in milligrams per milliliter. I also have log P, log D, and then is it a substrate for 1A, and then what's the predicted IC50 or PIC50 in her. So this is just an illustration of some of the properties that can be shown in Admet Predictor, or in Medchem Studio, obtained from Admet Predictor. Okay. So in this next section, I want to discuss Admet Risk, which is our Admet liability score. So this is a flexible scoring system that encompasses many components of risk. So here are the 3 components in Admet Risk. So they're broken up into absorption, cytochrome risk, and toxicity. For each one of the properties we measure, you get a score of 0 or 1, 1 indicating that is liability for that particular property. So by default, we have 24 rules, and these are based on thresholds of the World Drug Index. So we performed a similar experiment that Lipinski did to create his rule of 5, in that we created a subset of the World Drug Index, then we calculated the properties for all the molecules, and then we set a threshold so that 90% of the commercial drugs would pass this threshold. So if you get a +1 score for any one of the properties, you're falling within the 10% of the commercial drugs. So this is an easy way for chemists to see in rapid dimensions or many dimensions with a single number. We also augment the risk with a 2-letter code to identify which rule has been violated. This also allows a rapid way to compare and filter very large numbers of candidate compounds. So what we did was we calculated these Admet risks in Medchem Studio and then filtered in order to come up with our candidates in the Intimolaria project. Okay. So this slide simply on top illustrates how Lipinski defined his World Drug Index, or subset of the World Drug Index. So he started with a 1997 version of the World Drug Index. We started with the 2007 version, and we ended up with 2,300 compounds in our subset of the World Drug Index. So this illustrates an example of how we calculated the cutoff. So here we have log of the volume of distribution. We simply calculated all the properties, and then anything above 5.5 falls into the 10% of let's say, boundary, the excess boundary for the drugs. Okay, so the next couple slides show what the Admet Risk rules are and the code. So this is our absorption risk. So these are going to be similar to Lipinski rules. The first one is size, so you see things like molecular weight, or number of atoms, or the volume, number of rotatable bonds, hydrogen bond donors, acceptors, overall charge of the molecule, lipophilicity, permeability, and water solubility. And then down here are our risk rules for pharmacokinetics, so percent unbound in plasma protein, and then volume of distribution prediction. So if the volume of distribution is above 5.5, it would be flagged. This slide illustrates this SIP rules. So mainly we're looking to see, is the compound cleared quickly, which obviously would indicate an Admet risk. And then we also look at inhibition of metabolism, midazolam, and testosterone. And then finally, our Admet Risk rules for toxicity. So we have a hERG rule, rat acute toxicity, rat and mouse carcinogenicity, liver adverse effect, hepatotoxicity, and then finally Ames mutagenicity. So let's just look at a few of these examples again in Medchem Studio. So again, we look at the subset of about 1,000 compounds from GSK. So this top row here, the absorption risk is 2 for this particular compound. The code is octanol water and water solubility. The predicted log P is 5.3, so that's on the upper bound, similar to Lipinski's rule. And then the water solubility is only 2 micrograms per milliliter, so this is predicted to have low solubility, and therefore it's flagged. If we look at this third row, this is being flagged as hydrogen bond, too many hydrogen bond donor groups. As you can see, we've got one, let me make that a little bit bigger. So we've got 1, 2, 3, 4, 5 hydrogen bond donors, which would put it up in the upper 10% of compounds in our subset of the World Drug Index. And then finally, this compound was flagged because of its permeability, and it's has low permeability, 0.073. Okay. So that kind of illustrates the Admet Risk rules. So going back to our overall design strategy, the next step is to take these 13,500 compounds and classify them or group them together so that compounds contain a common substructure. And we use Medchem Studio to perform this task. So what our class generation algorithm in Medchem Studio allows you to do is automatically find common structural elements in a very large number of compounds. Then once the classes are created, you can use this as a data mining tool. So it will simplify the analysis of high throughput screening data. You can also use it to develop a knowledge base for what makes a good inhibitor of this enzyme, etc. So it shows how different substructures relate to properties and how specific constituents around a scaffold can affect various properties. So just to illustrate one of the methods used to create these clusters, I'll illustrate the ring system method. So if you take a compound like this amide here, the first step is to partition the molecules into rings and connective rings. So typically what I say defines it needs to have a minimum of 2 rings in order to be in a class, or as a scaffold in a class. And so it would take this molecule and it would classify it into these 3 groups. So here's the isoquinoline ring, a phenol linked to a purity by a 2-atom spacer, And then finally, the third class is the phenyl ring, a 2-atom spacer, and then the isoquinol ring. The next step is to take each one of these ring structures and append functional groups. So it would take this middle one and add the carbonyl oxygen. In another step, it would add the hydroxyl OH group, and then finally combine these two to make these. And then these three would be candidates for scaffolds, and then so we'd search for other molecules with similar scaffolds and then create classes based on those. Okay, so let me just flip over here and demonstrate class generation again on our subset of compounds. Okay, so here again we have the GSK subset of about 1,000 compounds. So here in this column is the activity column. So this is the XC50 that was calculated, our experiments from GSK. We actually created two DHODH models, and they're illustrated as Model 1 and Model 2 in this. We also have Admet Risk. One thing I haven't talked about is a star plot. A star plot allows you to plot up to 16 different properties within the spreadsheet. So what I've illustrated here is a wedge for the molecular activity. And if you hover over the wedge, it shows that the activity in all these 1,000 molecules varies from 4 to 8, and that this molecule is 6.23. So the length of this wedge relates to the value. So here's the least potent compound as far as this activity. Here's the most potent, so it's filling the wedge out. Similarly, I've got wedges for the pKi of Model 1, pKi of Model 2, and then finally the Admet Risk associated. We can also color code cells by various properties. So in order to create a heat map, so I'm going to color the activity, the risk, and then the two pKi values using a red, yellow, green type system. So if we sort on activity again here, the lowest activity is 4.7. It's colored red. The highest is colored green. So this is the most potent compound in the data set, and the various predictions for the models for these compounds. Okay, so let's go up and generate classes for these compounds. So I'm just going to select the ring system algorithm and then click OK. And now it takes these 1,078 compounds and classifies them into 18 groups. So that's that fast. And so the scaffold will be highlighted in red, or excuse me, in blue. So here we have a diaryl urea as this class that contains 401 structures. Then these columns now are the average values for the 401 compounds in this particular class. Again, and then the star plot is also the averages for these properties. We can also create individual distributions of the various properties. So here's the distribution of Model 1, the distribution of Model 2 for these 401 compounds, and then the Admet Risk for these compounds. So you can quickly see that there's a lot more risk associated with the compounds in this class than in the second class. Another thing we can do, so by default, the representative molecule is the lowest molecular weight compound in that particular class. We can specify that it be one of the more active compounds. So if we look at, again, our predictions from one of our models and click on the max button, now this is the most potent compound based on our pKi for that particular class. I can also define the pKi. Instead of being the average, I can set the function to the maximum. So now this value corresponds to the pKi for this compound. I can sort on pKi, and as you can see, you'll start to get clusters, or these three rows have the same most active compound but slightly different definitions of the scaffold. You'll see how we use that to select attractive classes within Medchem Studio, within our anti-malaria project. Okay, so now let's go back to the slides here. Okay, so now we've generated classes. The next step in the process is to identify attractive structural classes after we've classified them together. So what our process here is first to generate classes in Medchem Studio based on the scaffold. So that was just illustrated. Excuse me. And then find distinct classes that share very active compounds. So this is what I illustrated by sorting on the pKi, and then the representative was the most active compound. Examine activity distributions in the class, and particularly look for classes that have high predicted target potency and observed effectiveness in vivo. So this may indicate that these compounds are operating by inhibiting this target enzyme. And then finally, we want to look at the distribution of the Admet risks. So if a particular class has a large number of Admet liabilities, it may be difficult to improve the potency without increasing the risk. Now, if all the compounds in the class share a particular risk, then that may be due to the scaffold. So that may be a class that we want to draw. So this slide simply illustrates a little bit further what I showed in the demonstration. So we have the predicted classes here, the observed activity in vivo, and then you see that these two sets of compounds share the same most active compounds. So these are classes that we look into more detail. Okay, so once we've identified attractive structural classes, the next step is to generate novel compounds. So once scaffold has been defined, we can go into our R group pairs tab, and so it shows the molecules where you have a scaffold, and then the R1 groups in this case are the rows, the R2 groups are the columns, and then each cell either contains a circle or is blank. If it contains what we call a circle plot, I've colored these by the percent inhibition from the GSK donated data. So red is more of a lower potency, although it's 88% in this particular class of compounds, all the way up to 99%. And then areas where there aren't a circle indicate unexplored chemistry space. And so we'd like to enumerate these compounds. And that's very easy to do within Medchem Studio. I'll go ahead and illustrate that in this. So here we have a scaffold of azoindole, and then we have the R1 and the R2. I simply click on the R group pairs to come up with this screenshot that I showed on the previous slide. And then to enumerate these, I simply go under R tables, explode products, and basically what we're going to do here is combine every R1 group with every R2 group. Okay. If there's a particular R2 group that we don't want to see in the product, we can disable that very easily. We can also query for particular groups that we'd like to throw out, so query for them structurally, and then select those and disable them. So I need to specify an output file. So I'm just going to put something on my desktop. I'm also going to ask it to remove products that are already contained in this class. Then I simply click OK. It goes out and enumerates these compounds very quickly, and then brings up a new Medchem Studio with the compounds. So there's 184 compounds that exploded, and I'm going to select them all and combine them into one class. And then an easy way to define the scaffold is simply to select it kind of using a lasso tool. Now that's highlighted in blue, and that's what I've defined as the scaffold. I can go into the R group pairs, and then as you can see here that it's filled in with the circle of all the cells except for the ones that were already contained. So The next step would be maybe to calculate Admet properties, predict the activities of these compounds, and then search for ones that had maybe better properties, maybe predicted to be more potent than your lead compound. Okay. So now going back into the slides. So we identified a particular scaffold within the GSK data set, and it's illustrated by three parts of the molecule here, A, B, and C. At this time, we have a patent of the compound, so we can't disclose the structures, and so you can think of it as illustrated by this cartoon. So we identified a general scaffold. The next thing we did was to remove some very large and unusual substituents, and then we used the R-table explosion, and this yielded 142,000 candidate compounds. Then we predicted their activities with the model, we assessed their Admet risk, and then we filtered. So we threw out compounds that had a predicted Ki of greater than 100 nanomolar, and then anything with an Admet risk of greater than 5. This narrowed it down to 34,000 compounds. Then we applied an applicability domain filter. This reduced it further to about 28,000 compounds. And then we did some more filtering in Medchem Studio to kind of refine our selection of compounds. Okay, so we narrowed this down to say 20 or so compounds. We performed GastroPlus simulations to simulate the dose that eliminated a few more compounds, and then we sent these out to various contract research organizations to bid on the synthesis. Then we had to wait several months while the compounds were synthesized and tested in the biological assay. And our hope was that at least one of the compounds we made would inhibit the parasite at the molecular level. So once the results came back, they ended up being very nice. We ended up with 37 nanomolar hit compound. Excuse me. So these are the simulation plus IDs of the compounds. These are two somewhat close or analogs of our compounds in the GSK data set. Then here's the predicted DHODH activity of the compounds, and then the experimental growth inhibition assay results. So 10 nanomolar was the least potent compound, and 10 micromolar was the least potent compound, and 37 nanomolar was the most compound. The compounds were also tested for a drug-resistant parasite. As you can see, the numbers are a bit bigger here, so we're not quite as potent in the drug-resistant, but we feel like we could optimize these properties. We're still at 210 nanomolar. Okay. We also had these compounds tested for clearance by recombinant CYPs. So here in this are the 5 CYPs we had them tested in. Then our models predicted if they were substrate for these various cytochrome P450s. And as you can see, and then we predicted, we have the observed and the predicted clearances for each one of the compounds. As you can see, we predicted the clearance fairly well for most of the CYPs until you get down to 3A4. So here's where our predictions really fell off compared to experiments, and these were cleared fairly quickly by 3A4 actually. Okay, we also had physical chemical properties measured for these compounds. So this table illustrates our predicted value and the observed value for the 4 of the compounds here, and then the root mean square error for these. And these are all within our error estimates of the model, so we actually did a fairly good job of predicting these physical chemical properties. Okay, so to summarize, every molecule we designed and synthesized is an inhibitor of the parasite, so obviously that was a very nice result. We're not sure if the mechanism is inhibition of DHODH, but we think this is likely, and we're trying to get these compounds tested in this particular enzyme assay. The physical chemical properties were reasonably well predicted. The metabolism, unfortunately, was underpredicted especially for 3A4. The half-life is very short in the microsomes, but if the metabolites are active and non-toxic, this may be okay. Okay, so this finally just summarizes our discussion today. So started out with the strategy for designing the potential anti-malarial compounds, discussed building artificial neural network ensembles, and gave an example of building a PFDH or DH predictive model. And I think I illustrated that this is very quick and very easy to create this predictive model. And by no means do you have to be an expert modeler to create this model. Also discussed the 140 predictor models in Admet Predictor, talked about Admet Risk, which is our liability score, generated classes with Medchem Studio, and then performed an R-group explosion to generate novel compounds, and then finally discussed our results of testing and synthesizing and testing the compounds. Finally, I'd just like to thank my co-workers. Really all the credit for the anti-malarial project goes to Bob Clark, who is our Director of Life Sciences. We want to acknowledge GSK for donating the data to Pubchem. Collection performed the synthesis of the compounds, and they were tested in a biological assay at UC Riverside. And I'd like to thank you for your attention. So I'll turn it back over to John DeBell.
Thank you, Michael, for the excellent presentation. And we'll now open the webinar to a questions portion. Once again, if you'd like to ask a question using your telephone, please be sure to use the hand-raising icon on your control panel and enter your unique audio PIN. Or you may send your question directly to us using the questions pane on your control panel. And I do notice that there are several questions that have been asked. And joining us as a panelist is Robert Franskevich, who is our team leader of the Admet Chem Informatics group. And I know that Robert has assisted with the answering of some of these questions, so maybe he can help verbalize these responses so that the rest of the audience can appreciate them. The first question is, how does the volume of distribution greater than 5.5, that rule that's part of the absorption or pharmacokinetic risk, relate with the clearance of that compound?
Can you hear me?
Yes.
Yes, Robert.
So high volume of distribution usually indicates a significant amount of tissue binding. There is no direct relationship between total clearance and volume of distribution, but I can imagine that since a molecule is active onto the tissues, it does affect its transport properties back to blood, and that can slow down clearance. Hopefully that is the answer.
Yes, thank you, Robert. A second question is, does the GSK data set containing peptide bonds have a protease liability model? And if it is so, will your software predict or flag that?
Mm-hmm. So I can answer the second part of this question. I have to refer to Michael or Bob Clark and their knowledge of the GSK data set, whether it contains such compounds. Currently, we do not have a predictive model for protease. So the answer is no.
Yeah, I would agree with that answer. I think there are some amide bonds, obviously, in some of the structures that I showed, whether they're susceptible to peptidases, I can't answer. But I think that would be a good model to try and create.
As a practical matter, this is Bob Clark. As a practical matter, that the actives from GSK were all screened in blood culture. So they're active despite being exposed to all the proteases that are in blood, which represent a pretty wide spectrum of activity. So except for potentially specific proteases inside the intestinal tract, these particular ones that showed up as being very active probably are reasonably resistant to protease activity.
Thank you, Bob. Another question is, maybe it is because I missed the beginning of the webinar, and this asker does apologize, but it is not clear to me what data you have to initially know and put into the software to test your compound. Is it only the structure?
So in the context of this Admet design suite, all the predictions in Admet require only the structure, two-dimensional structure. For activity predictions, we have to have a data set of measured activities. And these actually can come from the literature, as we did with Pka Model.
Thank you, Robert. In the R group pairs plots, what does the green ball represent again?
That's just the most active compounds for that. So these spheres or circles are colored according to this activity down here. In this case, it was the percent inhibition at 2 micromolar from the GSK data. So this would illustrate a compound with around 99% inhibition. So these are more active compounds.
Thank you, Michael. On slide number 21, is the intrinsic clearance in microsomes, does it represent phase 1 only or phase 1 plus glucuronidation?
Phase 1. So I don't think that this would include glucuronidation.
Bob or Robert, do you have any additional comments to add?
No, yeah, it does not include glucuronidation. It's only SIPs.
Thank you. Staying along these lines, is the SIP metabolism predictions, are they based on crystal structure of enzymes or homology models, or can you go into a little bit more detail about these SIP metabolism predictions?
So I can answer that. The SIP metabolism predictions are based on two-dimensional structures and on a machine learning artificial neural network specifically, and models developed by us. That includes both the metabolic kinetics as well as the site of metabolism. So no structure, no crystal structures were used explicitly, although the nature of modeling, the technique that we use, has learned the active site of cytochrome dependence, it learned implicitly. Does that answer the question?
Yes, thank you, Robert. When using a cellular-based assay with no known mechanism, how did you identify potential targets and generate a list of known drugs that inhibit these two targets? Was this via your software or via Pubchem mining?
Bob, you want to answer that one?
What we did is used for the anti-malarial work. What we did is took a target where there was published data for compounds that were known to inhibit that target, built a QSAR model, and then used that model to pick out a structural class from the whole cell assays of the parasite that we thought was probably attacking that target.
Yeah, so this slide kind of illustrates. So we looked at cyclin-dependent kinase. So we obtained 46 compounds from the literature. Here's the 2 publications. And then the DHODH, one source was bindingdb.org, where we got a number of the compounds. And so when I illustrated the building the data set. These are all compounds from the literature, and their Ki values derived from the literature.
Thank you, Bob. Thank you, Michael. A next question: is it possible to import user-made descriptors to the software for use in model building?
Yes, by all means.
So going back to Admet Predictor, so it would just be, you would just add those descriptors as part of the files you read in. So if you had a Smiles file, an identifier, and then your proprietary, your own descriptors as a tab separated list, you would simply read that into Admet Predictor. And then when you build the model under the basic modeler setting tabs, your descriptors would show up on the left over here, and you would simply select those and then click this button to transfer them over on the right, and they'd be available for the model building.
Thank you, Robert. Thank you, Michael. Kind of going along those lines, if you do have some data, some experimental data that you've collected, for example, you did some microsome experiments, noted kinetics and extent of metabolism for some compounds, can you add the data to the model or models in the software?
Yes, absolutely. You can import the data as user-defined columns into Admet Predictor and use them as descriptors. Simply, they will show up in the left panel that is on the screen right now, and you would select those columns and transfer to the right for model building.
Oh, I think he was actually talking about experimental values he measures himself. So if you had something you measured, let's say a Km or Vmax, you could import those and create your own model for those.
Yeah, I understand. So I think the question was, can you replace predictions by measured values in Medchem Studio? Is that the question?
No, I think I
Then
interpreted
yes,
it.
I think that's the nature of the question. If not, please correct me. Then yes, I can imagine that Medchem Studio will accept measured properties as a replacement for predictions.
Okay. And then you also have the ability to utilize your own experimental data to extend the coverage space for some of our existing models as well, correct?
That is correct.
Okay, so I believe that might have also been maybe the nature of the question. Another question is, how well does Admet Predictor predict the properties, specifically physicochemical, of natural products? So
we have not tested the software on an exclusive database of natural products. It's an interesting question, and it's interesting to find out. I would like to point out that the program is available for evaluation at no cost. It will be a fully functional program running for a limited period of time, but there is plenty of time to put in a database of natural products with known measured properties and test predictions against the products. So you're welcome to try it. Anu will take any demo request.
Many of the models are trained on a much wider data set than simply drugs. In fact, most of them are. In addition, if something falls outside the descriptor space, then it gets flagged with the magenta coloring in the Admet Predictor spreadsheet. So some natural products will do that for some models, but you get a heads up indicating that it's probably out of scope.
Thank you, Robert. Thank you, Bob. For the permeability prediction in inside of Admet Predictor, where you're incorporating a two-dimensional structure, do you have any information about the commonly seen outliers for the PEF model?
So yes, we do. The PEF model is, as you know, based on measured permeabilities in human. This database was obtained by Hans Lennermas and Gordon Amidon in 1990s, and it's a small data set. It features only 55 compounds that we could use to model, and frequently, or maybe not as frequently, but sometimes the new compound lies outside the applicability domain of this model. And so, the commonly seen outliers are proprietary structures of our customers. So I'm sorry I cannot discuss the makeup of these compounds.
And there are, in general, it does not take account of active transporters or of transporters at all.
Yes,
so things that are PGP substrates. Yes, PGP substrates will be underpredicted, will be overpredicted. Things that have active transport, in many cases, will be underpredicted. But so it is truly a passive model.
Thank you, Robert. Thank you, Bob. I believe this question was answered already, but if you could just clarify, if I want to use the Admet Predictor software, do I need structures with data to train the model, or can I just directly use the existing models to generate prediction results with structure only?
I think I need clarification on the question. If you are talking about predictions involving commercially distributed models, then yes, you only need input structures. If you want to add a new model of interest to the program, then you will need both structures and measure properties to build the model yourself.
Thank you. Thank you, Robert. Related to the P450 metabolism models that are in Admet Predictor, which enzymes, can you describe a little bit more about which enzymes would metabolize a drug and also at which positions? So what specific CYP isozymes have we developed models for predicting sites and also extent of metabolism?
Oh, okay, it's on the screen right now. In terms of sites of metabolism, we have a set of 9 enzymes. It's 1A, 2A6, 2B6, 2C8, 2C9, 2C19, 2D6, 2E1, and 3A4. So in my written response, I missed 2E1. I apologize. Michael, can you please bring?
Yeah. So, yeah. With it. Oh, you want the other screen? I'm sorry.
Yes. Yes.
Okay.
So for kinetics and clearance models, we don't have data for 4 of the aforementioned enzymes, and we have models only for 5. Fortunately, these are 5 major cytokines, so I should hit most of the situations. 1A, 2, 2C9, 2C19, 2D6, and 3A4. We also have models that we have mentioned, but we have models of phased metabolism involving glucuronidation. These are classification models for the UGT enzymes that are shown on the screen.
Thank you, Robert.
Yeah. Just to add to that, so within Admet Predictor, it's a little bit far. So if we go to the calculated the properties and then go to the metabolism and then click on a gray cell, it'll bring up another window which shows the molecule and the predicted site. So for this molecule here, this methyl group is predicted to be metabolized by 1A2, so it would hydroxylate in this position. We also can bring up Medchem Designer. So if we do a shift click from the Admet predictor, it will bring up Medchem Designer, and then we can simply click on the M button here, and it will generate the predictions for this molecule. So here it's predicted that 1A2 and 3A4 will both hydroxylate this compound, and then here's the predicted product from the isozymes.
Okay, one last question that we'll answer for the benefit of the audience, and then there are a few other questions that we'll follow up with individually with each of the askers. If one supposes that the cysteine protease as a plasmodium target, did the software filter off reactive species as unwanted? For example, such natural products in fact inhibit the cysteine protease from plasmodium. So essentially, can rules for risk and toxicity be changed by the user?
Yes, absolutely. All risk rules are user customizable. The existing rules can be modified. The new rules can be added. And as well as you could see, the other Twitter features fully functional model building engine that users can very efficiently use to add new models. That includes protease or any other enzyme they're interested in. They will be appended to the program, and then they could be just as any other column added to the Admet Risk exposure.
In addition to that, Medchem Studio allows you to search for any smarts query, and then you can use that to filter out any reactive substructures that you would be concerned about.
Thank you, Robert. Thank you, Bob. As I mentioned, there are still a few questions, but we're going to follow up individually with those folks with their questions. Anu, if you want to just mention briefly about where we'll be next, and also the contact information if people want to reach us for evaluation copies like Robert discussed.
Thank you, John. Yeah, so I would be happy to send you slides. Actually, I will be following up with all the attendees after this webinar, and we would be happy to set up 2 weeks of free evaluation licenses. So you've learned about Admet Predictor and Medchem Studio in this. However, if you would like to try GastroPlus and DTD Plus, so I highly recommend that you do, because it's one of our great flagship products. So please contact John Tabella for GastroPlus and DTD Plus, and for Admet Predictor and Medchem Studio, you can reach me, as you see with my contact is on screen. And if there is a particular project you have on the site, Michael would be happy to assist you with those type of services. And next, we will be seen at the upcoming CTAC North American 33rd Annual Meeting in Long Beach, California. So if you are attending this, please stop by our booth. We will be exhibiting from November 11 to 15 in Long Beach. And we are also holding a workshop on GastroPlus in Mainz, Germany. So for our attendees from Europe, we'd be happy to assist you further. You can meet us in Germany if you're in Germany, and the details are on our website. So this is a workshop on GastroPlus, three days a workshop with extensive training on the use of the software. And with that, we appreciate your time and your attention, and thank you so much for joining us.
I would like to use the opportunity to just correct one answer. I said that no databases are available from us, with one exception. Okay, we offer the biorelevant solubility database for anyone that is interested in solubilities in simulated intestinal and gastric fluids. So that's for getting about it.
Thank you, Robert, and also thank you, Anu. One last comment. Going forward, what we'd like to do is host more focused webinars. As you can see from today's presentation that Michael gave, our software programs address a variety of activities during the drug discovery process. And what we'd like to do is have more focused, detailed webinars. If there is a particular topic that you'd like us to cover, please do send your ideas over to us, either to Anu, Michael, or myself. That is going to conclude today's webinar. If you did miss any part of today's presentation, the playback will be available at our website, www.simulations-plus.com, and we will also make available the presentation slides as well. Thank you so much for joining us today, and have a wonderful day.